And while we're not going to be making things today with biotechnology, we'll be using some of the tools of biotechnology to analyze what's happening inside of cancer cells, for example. And there's some really amazing technologies that are out there that we're going to spend some time going through. Today's lecture won't be quite as manic as we've had some of the ones in the past. So I won't be pulling my hair out anymore because I don't have any more hair to pull out. Last time I finished with agarose gel electrophoresis, and I pointed out to you that you have a support, which makes it sort of a mesh, and that's that molecule I showed you. That's a polymer. And this leaves little pores in which the buffer actually resides. The reason that gel electrophoresis techniques work is because you have molecules that have a constant size to charge ratio. And so the bigger they are, the more charge they get, so the ratio stays the same. And so when you force them through this mesh that I described, they separate on the basis of size. And so inherent to that, for the kinds of things that we're talking about here, the molecules must be negatively charged, and the molecules have to be rather rod-like in shape, like DNA is. So DNA works really easily. With DNA, we have a phosphate backbone. It's rod-shaped, okay? And so it has that constant size to charge ratio. And the negatively charged electrode near the wells pushes away the negatively charged DNA, and the negatively charged DNA is attracted to the positively charged electrode at the other end. So we get a gel like I showed you one last time. Here's another one this time. This gel looks a little bit different than the one last time because this one has been stained with a dye called ethidium bromide. So ethidium bromide is a molecule that when it's bound to DNA will fluoresce a sort of an orangish pinkish color, and that's what you see on the screen here. So in order to visualize DNA, I have to some way mark it, stain it, and that's exactly what's happened in this slide. Well, DNA is fairly easy to understand, separation in agarose gel electrophoresis. We can also separate DNA by PolyAcrylamide Gel Electrophoresis, and this uses a different mesh support. It's a chemical support, polyacrylamide. So acrylamide is the repeating material. And then these acrylamide strands are cross-linked using a reagent called BIS, B-I-S. So you can see the polymer on the left. You can see the cross-linking agent on the right on the image up here. And like we had with DNA, you might say, well, why do we use polyacrylamide versus agarose gel electrophoresis? Well, since the polyacrylamide has smaller pores, smaller holes through which things can move, it's better for isolating smaller molecules. So most DNAs will separate on agarose gel electrophoresis, but if we're looking at very small DNA molecules, polyacrylamide gel may work actually a little bit better in terms of being able to separate things. So it depends on what size of things we're trying to work with. Well, the other thing that we try to separate that's a macromolecule on polyacrylamide gel electrophoresis are proteins. So proteins are much smaller than nucleic acids, as I noted in class last time. And the proteins are, on the surface, it seems like very easy to think about, but the proteins actually have two properties that pose problems for us for separation. One is they're not evenly charged like DNA is, because DNA has a phosphate every nucleotide that's added. And protein charge, as you've seen, varies from one protein to the next. One protein might be positively charged at the pH of the buffer. Another one might be negatively charged at the pH of the buffer. So if I want to use the trick that I used to separate the DNAs on the gel electrophoresis, then I have to somehow make that protein be like the DNA. And so this turns out to be a very cool trick that we use for separating proteins. The cool trick is we take our mixture of proteins. So imagine we've busted open these cells. We've got all these proteins that are sitting here in this tube, and we want to separate the proteins by gel electrophoresis. Okay? So the way that we would separate them would be to mix the proteins with a detergent called SDS. SDS stands for sodium dodecyl sulfate. As long as you know it as SDS, you're fine. Okay? And by the way, any abbreviations I use in the class, you are welcome to use. you have to get the abbreviations exactly the same. Like you can't call SPS, for example, and say, well, I had 2 out of 3 letters right. All right, you got to get the abbreviation right. But you're welcome to use any abbreviation I use in class. Okay, so SDS is treated with these proteins, and what SDS does is a very interesting thing. First of all, SDS is a detergent. So if I said detergent with protein, what's the first thing that you should think of? Denature, right? And so globular proteins, that's the other reason proteins are hard to work with. Remember with DNA we had rods? Globular proteins are all folded up in balls. So if I denature those proteins, what's going to happen is that they will actually stretch out and be rod-like, number one. Number two, this may surprise you, but it happens. The SDS arranges itself as a sort of a shell around that protein rod. The nonpolar part facing the polypeptide chain, and the negative charge, SDS, like most detergents, is negatively charged at one end, the negative charge sticking out. Well, what have I just described to you? I've just described a rod that's coated with a negative charge. And the longer the protein is, the more SDSs I'll have, and the more negative charge I will have. So just like the DNA molecule had more negative charge the larger it got, so too will most globular proteins have more of a negative charge the longer they get. So in essence, the proteins are all going to have the same ratio of size to charge. Now you might say, well, what about the charges inside of the protein, right? What about those charges? Because proteins have their own charge. Those charges are usually on the order of 1, 2, 3, 4, 5, 6, maybe 10, maybe 20 at most. And we're coating this thing with hundreds of SDSs. So we don't even really see the charges of the protein itself. We see the charges, the gel sees the charges of the coating, which is the SDS. Everybody with me? So now we've artificially created rod-like molecules with the same charge, size-to-charge ratio. So, like before, we can use the same setup that we had, except for now we're using something that's got smaller pore sizes, this being the PolyAcrylamide Gel Electrophoresis. And by the way, we call this technique, when we add the SDS, we call it SDS PolyAcrylamide Gel Electrophoresis, which is a mouthful. You can call it SDS-PAGE, P-A-G-E, PolyAcrylamide Gel Electrophoresis. So when you hear SDS-PAGE, you know the sample has been treated with SDS, okay? And it's done that coating that I've described to you here. Well, again, all molecules have the same mass-to-charge ratio, so it's going to act like the DNA molecules are going to act. It's going to separate on the basis of size. The largest molecules will move the slowest because they have the hardest time getting through those pores. And this shows a separation of a protein, a set of proteins, and both the figure on the left and also in the figure on the right. They've been stained with different things. But suffice it to say that you can isolate, obviously, all of the proteins in a mixture, or separate the proteins all of a mixture like this. One of the questions you might have is, well, if you've denatured this protein, you try to take it out of the gel, you know, is it going to be any good? The answer is usually not. Usually what you're doing with this technique is you're usually trying to see what are the sizes of proteins that you have in there, and you may have other analyses that you'll do of them. But you're usually not trying to get functional protein out of this separation. Okay? Largest at the top, smallest at the bottom. Okay. Well, polyacrylamide gel electrophoresis. Question? Somewhere. Yeah. Yes. Yep. It does repel more. But it's the ratio of size to charge. Because the ratio of size to charge is constant, then the only separation basis is size. Just how well they can get through that. Because they have the same net of force that's being applied to them relative to their size. Okay? Good question. Other questions? Okay. All right. Polyacrylamide gel electrophoresis, blah. Oh, I've got to go through all that crap. Okay. Isoelectric focusing. Now, this is a technique that's a little hard to get your head around, so I'm going to try to put it in a little simpler terms than I usually do. Okay? So isoelectric focusing, first of all, I'll tell you what it does. It's a way of separating proteins or other molecules, though almost always it's used for proteins. It's a way of separating proteins on the basis of their pI. How did I define pI in the class earlier? What's the pI? So it's a pH, first of all, right? What pH is it? Well, that's how we calculate it, but I'm saying, what is it? What pH is this? Why is this pH significant? It's a pH at which a molecule has a charge of 0. Remember that, okay? People say, it's where the molecule has a charge of 0. No, it's the pH where a molecule has a charge of 0, right? All right. So it's the pH at which a molecule has a charge of 0. Remember, we're scientists. We need to be precise, right? pH at which a molecule has a charge of 0. So this technique separates molecules on the basis of their pI. How in the world does it do that? Okay. Well, let's take a look at the strategy. You see we have a column, just like we had before, but this column has a material in it that's sort of a gel-like material. Okay? You can think of it as beads if you want to. I think of it as a gel-like material, but whatever works for your head works for me. And that gel-like material that's in there has thousands, millions of different beads that differ in charge. Some of them have a lot of negative charge. Some of them have a lot of positive charge. Some of them have a charge close to 0. Some of them have a charge of 0. Okay? Now, if I apply an electrical current to that column, what's going to happen to those beads? Well, the most positive ones are going to go towards the negative electrode, and the most negative ones are going to go towards the positive electrode, and the most 0 are going to go towards the middle. Right? I'm not talking about separating any molecules. I'm talking about the material in the column. Well, what that does is that establishes a pH gradient, because those guys have protons associated with them. So what I've made is a column that has, let's say, a high pH at one end and a low pH at the other end. And no, I don't care if you know which end is which. But you know that you're setting up a pH gradient in that tube. Everybody with me? Let's say bottom in this case is 0, top in this case is 14. It's a pretty wide range, okay? Now, I apply my proteins in there and I turn on the current. What's going to happen to the proteins? I haven't treated them with SDS now. These are just native proteins, okay? Well, if a protein starts out with a positive charge, it's going to be attracted towards the negative electrode. And it's going to get to a point where the pH is going to cause it to have a charge of 0. And when it gets to that point, it's going to stop. Because at a charge of 0, there's no attraction for either of the electrodes. Things that have a pI value close to 14 are going to be up in the 14 range. Things that have a pI value down around 2 are going to be way down here at the bottom near the 2 range. So at whatever place they reach a pH where they have that charge of 0, they will stop migrating. Okay? Does that make sense? Now that's what this figure is trying to show you. I hope I've told you in words how that works. But what we see when we do this is we see a series of bands, each one corresponding to a unique protein at a unique, I shouldn't say unique because we may have a mixture and maybe a lot of things that's in there. But if we look at, let's say, pI of 5, we would have all the proteins at that band that had a pI of 5. Okay? Now, understanding this technique is important because it's actually used with another technique to make a really powerful analysis. So before I go and talk about that, I'll ask if there are any questions. Questions about this technique? Anybody awake? Yes, question. Very good question. Does it work better than the gel or the same? This is a little trickier than a gel, but the advantage, this would be a good exam question. If I said to you, what's the advantage of this technique compared to the SDS polyacrylamide gel electrophoresis, what would you say? There's something very different about these two in the way I've described them. Anybody? Stay intact, meaning the proteins do not denature, because we haven't added SDS. So if I wanted to separate on this basis and try to recover proteins that still were active, I would have a greater chance of this giving me that, right, than the other method. It still might not be active, because remember I've changed pH drastically, so I don't know that. But I would have a better chance because I don't have SDS in there. Okay? Now, so getting back to her question, is this better or worse than polyacrylamide gel electrophoresis? And all these techniques depend on what you're trying to do. All right? Polyacrylamide gel electrophoresis, or SDS-PAGE, would not tell us anything about PI. It would tell us only about size. This technique will tell us about PI, but it won't tell us anything about size. Did I say it backwards? I said it right? Okay. I forget what I said. All right. So we get no information about size on this, whereas the other method we get size but no PI value. Okay. So there's no better or worse in terms of that. Okay? Well, what this technique is really useful for is the power in combining it with another technique. Well, we did that last time. All right. All right. So what I'm getting ready to describe to you is a method that we are basically almost a discipline called proteomics. How many people have heard of proteomics before? A few. Okay. So proteomics is a way of analyzing simultaneously all of the proteins of a cell or a tissue. All of them. Okay? Now you might say, well, SDS-PAGE did that. And what you'll see is what I'm getting ready to show you is this is a much more powerful method than SDS-PAGE, because SDS-PAGE squeezes everything into one lane, and while we see a whole bunch of bands that's in there, we don't get very good quantitative information about each protein. The method I'm getting ready to show you gives us very quantitative and reproducible information about every protein that's found in a tissue, for example, or a cell. Okay, so how does it work? We use a technique called 2D gel electrophoresis. Now, 2D suggests 2 dimensions, and in fact, that's correct. One dimension of this technique is involved in using the isoelectric focusing that I've described, and the other dimension of the technique involves SDS-PAGE. We combine the two to make a very powerful separation technique. So let's start. We've got a mixture of protein, and we want to do 2D gel electrophoresis. So the first step of the process involves separating all the proteins in the mixture by their PIs. You've seen this. I apply the voltage. I get bands corresponding to that. In this case, I've got pH decreasing, so I'd have 14 at the top and I'd have 0 at the bottom. Okay? High PI, low PI. Okay. Next, I take that tube that contains that separated batch of proteins. I treat it with SDS, because remember I didn't use SDS before. I treat it with SDS, and then I lay it on top of a polyacrylamide gel that has SDS in it as well. Okay? And I do separation by polyacrylamide gel electrophoresis. This is the second dimension. Everybody follow? Separate by PI, separate by size, and what I do, and the first one, the top one's the first one, the down one is the second one. Okay? When I do this, okay, I see something that looks like this. Question, yes? So I treat, say it again. That's correct. And whenever I run SDS-PAGE, I've always got SDS in the gel as well. Yeah. So there's plenty of SDS there. Remember, we want to have these things coated as much as possible. If the gel didn't have that, then it would just come off, and I would see some unusual things. Now, what do I have? Well, I have now a gel that doesn't have just one lane of protein. The proteins are spread all the way across the gel. All the way across it, okay? Here is actually a real live, real world 2D gel. The things are separated on the basis of size in the up and down dimension, and they're separated on the basis of pi in the left and right dimension. So if, for example, the left side of this was the low pi, then things that are in the upper left will have a low pi and a large size. Things on the upper left. Things on the lower left will have, let me start over. I think I screwed that up. All right. Upper left are going to have large size, low pi. Lower left are going to have small size, low pi. Things in the middle, at the top, no charge or little charge, that is a pi near 7, okay? And large size. Middle of the bottom, going to have a low pi, middle, a pi around 7, and a small size, okay? You can figure out the rest, right? Now, the beauty of this technique is that on a single gel, I can separate these things so well that every spot corresponds to an individual protein. And not only does every spot correspond to an individual protein, but the darkness of the spot is a function of the quantity of protein. The darker the spot, the more the protein. So I can see there's some proteins there that have very little in the way of being present, others that are very abundant in terms of being present. And I get a relative analysis of those by just simply looking at the gel. Now, one of the things we learn in gene expression, and we'll talk about this next term, is that, first of all, not every cell makes every protein that it can. Second, not every cell makes every protein in the same quantity when it makes it. What you see on the screen is a representation of that. Some proteins are very abundant. Some proteins don't take as much. We'll talk about E. coli. That E. coli makes a DNA polymerase, for example, for which there's about 5 or 6 copies per cell. That's not very many. By contrast, ribosomal proteins that are present in E. coli can be present in millions of copies. Okay, so there's a big difference then in the amount of protein that's being made in any individual cell. All right. Just said that. I like this one. That's pretty cool. This gives a little bit better feel for what these kinds of analyses actually give you. If you were to sit down with an instrument and measure this, you would see that this technique would allow you to separate on the order of 4,000 to 5,000 proteins very easily. Okay? Now you see every protein from that cell. Every protein. And you can use sophisticated techniques of mass spectrometry to determine the identity of every protein that's on there. So you know this protein that shows up on the lower right and it's very dark and abundant. You look at that and you say, yep, I know what that one is. That's ribosomal protein L21. Right? And I could run a similar gel for a different tissue and say, wow, I don't have quite as much L21 in this cell. Right? So the reproducibility of this is important, and that separation and analysis is also important. Well, I've just described to you something that's obviously a powerful technique, but it's even more powerful if we do some tricks. So let's do some tricks and see how this analysis could help us. Let's imagine that we have 2 sets of cells. I've got one set of cell over here that's a normal set of cells. Let's say it's liver tissue. And I've got a patient who has liver cancer. And the patient has had a tumor removed, and tumor cells were taken from that tumor, and proteins were isolated. So the normal tissue, I've got proteins isolated. The tumor tissue, I've got proteins isolated. Everybody with me? Okay? So let's take the healthy cells and we'll label those proteins with an orange dye. That's fairly easy to do. Take the proteins from the cancer cell and label them in blue. Okay? We mix those We do the separation, and now we ask the question, what does the gel look like, first of all? And think about what this gel means. As we look at this gel, tell me what your interpretation of this gel would mean. I see three things on there. I see blues, I see oranges, and I see blacks. What does orange tell me? So orange says that these are proteins that are made in healthy cells, but not made in cancer cells. Blue, of course, is going to tell me these proteins are made in cancer cells, but not in healthy cells. And black is when I mix the two. Now, the intensity of each of those, the intensity of the orange, tells me this is made very abundantly in healthy cells, but not in cancer cells. Intense blue, of course, the same thing, et cetera. Intense black says, hey, there's a lot of it, but it's made in the same quantities in both cells. So we get something that's sort of bluish orange, right? That would have more blue than orange, for example. We'd say, well, it's probably a little bit more in cancer cells than it is in healthy cells. But we're actually getting quantitative information now about the difference of expression of each protein between a cancer cell and a normal cell. This is an incredibly powerful technique. This doesn't have to be two cancer cells. This could be, let's say, one batch of cells that I have not done anything to, and the other is a healthy batch of cells that I've treated with a new drug I'm interested in testing. Is there a difference in the expression of the proteins between these two? This technique tells me very quickly, and not only does it tell me very quickly, I know the identity of each protein that's on there, and I can say, whoa, this drug caused this protein to go up in expression or to go down in expression, right? It's a really powerful tool for us to be able to quickly determine the effects of drugs on any of these things. We can take a cancer cell. Here's a cancer cell that's untreated with this drug. Here's a cancer cell that's treated with this drug. Whoa, look what this drug did to this cancer cell's protein, and this protein is pretty critical for this cancer cell to be alive. Right? Amazingly powerful technique, and you do it in an analysis in a couple of days. Okay? Pretty powerful. Okay. Questions on this? Yes? I'm sorry, I can't hear you. Yes. Yes. I'm not sure I understand the question, but the question, as I understand it, is does the SDS affect the analysis here because I'm denaturing the protein? Is that safe to say? Okay. The answer is no, because again, I'm not trying to get functional protein out of this. Once I've treated this thing to, for example, the dye, many proteins would be just completely inactivated by that in the first place. So I'm not trying to get functional protein out. All I'm trying to do is to get the proteins separated here. Okay? Question? SDS does not break proteins up. The only thing that's going to break up is if you have multi-subunit proteins. But SDS-PAGE would do that automatically anyway. So for example, if I put hemoglobin, it's a good question. If I put hemoglobin in SDS, what's going to happen? Well, it's going to come apart because it's going to completely denature into rods. So the question is, no two proteins have the same PI and molecular size. You'll see some that are pretty close, all right? And I can't tell you that absolutely every spot on there is unique. It's possible that some spots will have more than one that's there, but for the most part, they're unique. Yeah. Yeah, question. How what? How reproducible? Very good question. This method is actually pretty darn reproducible. But this thing that I've shown you right here is one of the ways of getting around irreproducibility, because you're directly comparing two things. So if I were to, for example, take and run a 2D gel of a healthy cell over here, and to run a 2D gel of a cancer cell proteins over here, and I tried to lay them on top of each other, I might have to do a little shuffling to get them to, because this one might have a slight difference in the way it run than this one did. But when they're run on the same gel, I don't have that problem. But they are, in fact, quite reproducible. So that's the good thing, is that you can say, okay, well, if I now know where, for example, that DNA polymerase is on here, then everything else will orient relative to that. I could say, well, above that's going to be this, and over to this side is going to be that. So it's a relative thing, but I still get reproducible separations. Yes? Okay. So the question is it possible to do a 3D? And the answer is it is. It's limited by your imagination. Seriously. Okay? So 3D separation would certainly be possible. Imagine how you might separate something in a third dimension. The analysis would be hard because you'd have a cube, basically, at that point. But is it possible to do? Absolutely. So there you go. There's your $1 million. Other questions? Yes? Oh, is there to simulate it? Well, a computer can certainly... In the
same way that supercomputers can model protein folding?
Sure.
Same idea.
Yeah, a computer could easily simulate this. That's not a problem. The computer can simulate it, but the computer's not going to have any knowledge about relative amounts, and the only way you're going to get that is by analyzing an actual set of cells or tissues.
That's not just data you can input to the system and then it would run that, and even do it in 3 dimensions too.
Okay, so very good question. So if you input the information for each protein that you thought it would make in terms of quantity, the computer can simulate this without any problem. Computer simulations of gels, for example, are routinely done to predict what, for example, DNA a pattern might look like. Yeah. But generally, what you want is not the simulation when you're talking about a tissue or something. Is you want to see actually what's going on there. But you're right. You couldn't do that. Yeah. I'm sorry. I didn't mean to interrupt what you said.
Oh no, that's kind of what I was.
Okay. Yep. Good question. Yeah. Yeah. See proteins what? So is it possible to see protein cleavage? Well, you're not going to see cleavage unless you use a cleaving reagent. SDS is not going to cleave anything. But let's say you took and you treated your sample with a protease that chewed up proteins. Oh, you'd see a very different pattern here. Yeah, you bet. So again, because once you've altered the protein, you are now going to see a difference in mobility, both in terms of size and probably in PI as well. Yeah. One of the things that this is useful for is looking at modifications of proteins. We've talked about things like hydroxylation of proline and so forth. Well, that one doesn't show up as too different on an analysis like this. But one kind of modification shows up very readily, and that's phosphorylation. Because proteins get phosphates put onto them, and that's commonly used to control their activity. So if you would expect, well, you start putting phosphates on there, you're going to change that mobility a little bit because you're going to change the charge a little bit, right? And it turns out that actually can be detected by this method. So it's useful for looking at modifications that affect charge. Okay. All right. Black, blah, blah. All right. Well, we've seen now one way of analyzing, this is proteins at a level of the entire tissue. What I want to show you now is a way of analyzing the amount of messenger RNAs in an entire tissue. Okay? This one involves knowledge of complementarity of nucleic acids. So they're called microarrays. And microarrays are something that we make in the laboratory for analysis. And let me tell you how the process works. So first, in order to do this, I have to chemically synthesize a DNA that's complementary to a messenger RNA that I want to analyze. So let's say I'm interested in the messenger RNA for DNA polymerase. I have the complete genomic sequence of an organism. So I go and I look up and I say, okay, here is the sequence of this DNA polymerase. I go to a machine that will chemically synthesize the DNA complementary to that messenger RNA. Everybody with me? I've just chemically synthesized this DNA, and it's one strand, not two, one strand. Okay? So if I were to put the messenger RNA and the DNA together, they'd form base pairs, and everybody'd be happy. Okay. I take a grid, okay, which is just basically a glass slide, and I divide it into spots, like you see here. For what I just described to you, I take that DNA that I made and I bind it to the spot number 1 in the upper left. So spot number 1 will have thousands of copies of the DNA complementary to the DNA polymerase. Everybody with me? Each spot on this sample will ultimately get a unique DNA complementary to each mRNA. So, for example, 1 spot per mRNA. There's the first one that I put on. The second one goes on here. And that second one is going to have thousands of copies of DNA complementary to another gene, another messenger RNA. So I repeat this for every spot on this grid. And I keep track, of course, of which complementary DNA I've put onto each spot. When I'm done, I will have a grid that will have 1 complementary DNA for each messenger RNA of the cell, if I make it big enough. Yes? I'm sorry? On each spot is the complementary DNA that I made to the messenger RNA. So I haven't got the messenger RNA in the picture yet. That'll come later. But I know from the genomic sequence, the sequence of each messenger RNA, so I've made a complementary DNA to it. So this grid has complementary DNA on it. Everybody understand? Other questions? Okay. Well, question over here. Yeah. Just shout, yes. They are actual spots, as it were. Yeah. So it's a grid, and you can tell, okay, here's, 5 across and 3 down, and I know what that one is. All right, each one I know. All right. So now we're going to get to the messenger RNAs. I got 2 batches of cells. I've got a healthy batch of cells, and I've got a cancerous batch of cells, just like I had before. I isolate the messenger RNAs from each of those cells. That's easy to do. Okay? I take then and make a copy of the messenger RNA because it's easier to work with DNA than it is messenger RNA. I make a DNA copy of each messenger RNA. Okay? And I put a tag on it. Okay? The healthy cells get green. The cancerous cells, in this case, get red. Okay? I take those and I mix them. I don't keep them separate. I mix them. And I take that mixture and I apply it to the entire grid that I had before. Okay? I allow hybridization to occur. This is where complementary strands will find each other. And I've made this so that they're complementary. All right? And then I wash away anything that's not hybridized. So anything that's complementary to each of those spots is going to base pair with it, and then the unhybridized thing is going to go away. All right. What does it look like? Well, when I do this analysis, I see something that looks like this. I see greens, I see reds, I see yellows, and I see blacks. And I see others that have different shades in there. All right. The shade of the color is, again, a function of which thing is present. So, for example, bright green will be abundant in healthy cells but not in cancer cells. Bright red will be abundant in cancer cells but not healthy cells. Bright yellow will be abundant in both. And black will be absent in both. So now with this analysis, I can tell what is the amount of messenger RNA that was present in each of those tissues. Okay? Questions about that? A lot of steps in this one. Yes, Spencer. Are the microarrays reusable? They are. Yep. They're expensive to make, so you want to reuse them. And by the way, here's a real-world microarray. That's awesome. I'd say it's about 9 feet by... They're typically done on a slide. They're not very big. You have to analyze these under a microscope. Okay? Under a microscope, yep. That would be one microarray right there. So that whole thing would probably be about like that. Yeah.
So, is this analyzed by a person or a program?
Oh, good question. Is this analyzed by a person? I don't want to have the job. Well, it's very yellow, or is it sort of orange, or I don't know what that is, okay? The beauty of this is when we get into very high levels of data, big data, as you always hear about, that we have computers that do this analysis. Now, the computer knows the identity of each gene on that microarray, okay? It knows the identity of each one. And then the computer just simply looks at this, and it actually is able to quantitatively tell how much how much green is there, how much red is there. And it'll tell, is it favoring one versus the other? And it's very sensitive, way more than our own eye is sensitive. So it can tell us very good quantitative information about the relative amounts of each of those. And since it knows the identity of each protein, it can give a profile of that cell. You might tell the computer program, okay, what enzymes of glycolysis, are they affected by this? And so the computer program would go back and it would say, okay, here are the enzymes of glycolysis, here's the information, and you could see that 3 of the enzymes of glycolysis are elevated in the cancer versus the non-cancer cell. All right? Instantaneous information that the computer could pull out for you and tell you about the relative abundance of these. Okay. Yes? What's favorable about this over the 2D gel electrophoresis? I'm glad you asked that question. It's a different it's a different analysis. So we're measuring messenger RNA here compared to measuring protein there. Next term, we'll talk about gene expression. And gene expression is a function of many things. Simply because a messenger RNA is made doesn't mean that a protein is made from it. So you might want to know how much messenger RNA is made compared to how much protein is made. And if you see a big difference between those, you'd say, whoa, there's something inhibiting the translation of this protein. So both analyses are really important in understanding the phenomena of gene expression. But the answer to that question will really have to wait until next term. Other questions? I told you it'd be a little slower pace today. Okay, so these techniques are very, very powerful. They're very commonly used in laboratories now, and amazing information that you can get. Okay, this analysis is what people would call transcriptomics. Okay, transcriptomics. So that one was proteomics. A transcript is what a messenger RNA is what a messenger RNA is. We've heard about transcription, right? So this analysis of analyzing all of the messenger RNAs of a system is known as transcriptomics. And you might say, well, we're not measuring messenger RNAs, we're measuring those complementary DNAs, but the complementary DNAs were made from the messenger RNAs. Okay. All right. We have 2 songs to finish today. One relative to this, and one relative to, I realized we hadn't sung one on amino acids, so we'll come back to that one. Neither of these are recorded, so I'm going to have to lead these guys, and I'm very sorry for that. This is called I've Just Run a Gel. It's to the tune of an old Beatles song called I've Just Seen a Face. I've never sung this song in class before, so you guys are going to get this full blast. Please join me because I need help. I've just run a gel. I do not think it went too well. I may have used a bit much SDS. The stacker's looking like a mess. It's true. Oh, now what will I do? Protein samples, my last one to purify. It was not fun. I spent 3 weekends working late. The middle lanes aren't looking great. I'm screwed. Good God, what will I do? Crawling, I'm almost bawling. The boss is calling to follow through. I've just loaded all I've got to make this final Western blot. My fingers are both crossed for sure. I hope my protein product's pure, I do. Then my thesis is through. Hating all of the waiting, I'm contemplating, what should I do? Staining, my eyes are straining, there's no complaining, I say wahoo. because it has the band I need, I'll go and have it scanned to speed the writing of my thesis and proceed on to the postdoctoral plan. Oh, that will be so grand. Pieces make up my thesis, no more phoresis, the promised land. Writing so unexciting, but no more biting my nails again. Writing is coinciding with reference citing. I'm at the end. Okay, so that ended happily. The other one was a song about amino acids that we didn't get to sing, and I didn't even realize it until today. And this one's kind of fun. It's to the tune of an old Ringo Starr song called You're 16. And it goes, You're 16. If you've heard this one, please help me with this. You're in every protein that I've ever seen. There's no need to debate. You're cysteine, a building block, and you're great. You give the hair waves, it's something we crave. Makes the food on my plate. You're cysteine, a building block, and you're great. That sulfhydryl in your chain can oxidize, but I don't complain. It gives support to all peptides, so proteins need to have disulfides. You're an acid, it seems, and have an amine. Please don't ever mutate. You're cysteine, a building block, and you're great. How does this go? A UGU or UGC, it's secret code at the cell's decree. You're in my skin and in my bone and even in my glutathione. You're in every protein that I've ever seen. There's no need to debate. You're cysteine, a building block, and it's Friday. So it was, okay, so red and green, all right? We'll either have red or green. If you combine them, you get yellow. So yellow comes from the combination of the two.
Okay.
You put red and green together, you get yellow.
Oh, so the blockers are neither of them bonded.
Black means nothing's there.
Okay.
Yeah.
Okay.
I may not have been clear about that. I'll clarify in the highlights, okay? Come on over, guys.
Mine's regarding Wednesday's lecture.
Okay.
I was curious if the video worked or not, or if I didn't have the time to put the video up.
Posted it this morning.
Oh, you posted this morning?
Yep.
Okay. Thank you.
Yep.
I was still a little confused about the block, how it's run.
Uh-huh.
So, first,
What are we talking about?
like the color, the block.
Which one? Are you talking about the analysis of the messenger RNA or the analysis of the protein?
Messenger RNA? The one that's colored. That's the
One was orange and blue, and the other one was red and green.
Oh, they're two separate. I got this. So the messenger RNA.
Okay.
Do you add the